All right, so what we're going to do now is look at the chloroplast structure itself, and then the photosystems and how they all connect and what's going on with them with the process of, we talked about last time, photoexcitation to get the energy. All right, so this is going to be the main conversion process of the light energy into the chemical energy in the form of electrons that'll be stored in some molecules we talk about along the way. So, bigger picture, kind of putting it together. Light coming from the sun, that's our energy source. It's coming in a variety of different wavelengths. Plants can use the visible light spectra, wavelengths. There are a variety of different wavelengths that can be used by a variety of different types of pigments, each one absorbing light at different specific wavelengths, which you can get into in specific details, but we're not necessarily doing it. I'll talk about some of these pigments, accessory pigments versus the chlorophyll pigments a little bit more, but in general, just know they're kind of absorbing all this to get the energy. But the idea is, where is that happening? Okay, so within a leaf, say for a larger plant, if it's a pine tree, for example, the needles, those are the leaves. And the broadleaf plants and grasses and all, we're more familiar with those types of leaf structures. Some single-celled organisms that are photosynthetic. The whole cell, is this living organism itself. It's just a single cell that's carrying this out. But inside then a cell, so a plant tissue is made up of cells, and each cell has a nucleus. So we have the nucleus in the cell. So we blow this up. So this is like a piece of tissue with a whole lot of cells arranged there. You might see this under a microscope when you look at plants, leaves and roots, kind of recognize some of that. If you look inside the plant, they have a nucleus like all their cells. They have other organelles and endoplasmic reticulum. They would have mitochondria inside their cell with the same structures that we would have in our cells and so on. But then they're going to have the chloroplasts, which are more unique. So it's a single cell. So this is a single cell. And inside it, and even this is the same for a single-celled organism as well. So we're going to have the chloroplasts, and there's going to be a lot of chloroplasts. Just depends on the cell how many they may have, but they'll have a lot of chloroplasts. So a chloroplast itself is going to contain, its own membrane. So, and that membrane is going to be just be referred to as the outer membrane. Okay, so we have an outer membrane. Now, beyond that, remember mitochondria, they have two membranes. So chloroplasts are going to have at least two here, we're going to get into. They're going to have a second membrane that we would refer to simply as the inner membrane. Okay, so an outer membrane, inner membrane. For mitochondria, that's all there is. They're the two membranes. They're going to create two spaces at this point. There's an intermembrane space in the mitochondria. That space was very important. Here it's not going to play that significant of a role. We have this space to the interior. That's going to be called the stroma of the chloroplast. And then what we are going to have is a third membrane. And this third membrane gets folded into a whole bunch of separate structures, like little stacked pancakes, okay, like this. And that third membrane is called a thylakoid membrane. All right, important because it's one you probably haven't heard of it before unless you've studied this. And this is where everything's going to happen. Thylakoid is going to be where all the pigments are actually located. So when we say light is absorbed by a plant and it's absorbing certain wavelengths of light by these pigments, and some of the wavelengths are reflected, back off by these pigments. So we ask, where are the pigments located? They're not in the outer tissues here. They're not in the cell wall, all right, or the cell membrane. They're not in the cytoplasm of the cell. They are in the actual chloroplasts. That's where we find the pigments. The chloroplast now has all these different structures. So where do we find the pigments? Well, they're not in the outer membrane or the space between them or the inner membrane or that space inside them there. The pigments are actually embedded in these membranes here in the thylakoid membranes. So the thylakoid membrane forms stacks, these little stacks of these little compartments called grana. That's kind of the overall structure. An individual thylakoid, little compartment like that, then has a space obviously within it, okay? And that space within it is referred to as the thylakoid space. And that's important because that's also where a whole lot of things are going to happen. So activities that are part of photosynthesis are going to happen in the thylakoid space, between the thylakoid space and the stroma. Okay, so it's going to be that back and forth. So that's what we're going to mostly be focused on. Now, what I'm going to do here is now take a lot of blowing things up to get down at a finer level of detail. If we look at an individual membrane, so if this is a membrane now of a thylakoid, so this is going to be a thylakoid membrane. This is the membrane here. This is the thylakoid space inside it. So the thylakoid space, this would be the stroma here, and that's the membrane. Now, embedded in this membrane, we said first off are going to be the pigments. Now we said there's a whole variety of different types of pigments. So these pigments will be carotenoids, xanthophylls, the chlorophylls, and others. Other accessory pigments. Okay, so they're all going to be located in the membrane in an area we refer to as the antenna. Antenna array. Okay, and I'm putting number 1 here by the antenna array, because the antenna array is going to really be the first part of a photosystem. So what we're talking about now here is how does the light energy get sort of transformed into the chemical energy? And we said it's going to be happening through a process called photoexcitation, all right? And it's going to have to do with the pigments. What happens here in the photoexcitation process? Kind of pull this away so we have this overview of the structures that we're talking about. So we're kind of inside the cell, in an individual little organelles within the cell called the chloroplasts, and we're focusing on a particular membrane within that structure called the thylakoid membrane. And in that membrane we have all these different pigments, which you could look up. I have some in the your PowerPoints and in other places. We have references to the actual chemical structures of the pigments. For our class, you don't have to know that. Not biochemistry class, you may go into it in more detail. If this is just a pigment molecule, what's going to happen to this molecule is that as the light energy hits it, electrons in certain places, certain elements within this, are going to get excited. Which means they jump up to a higher energy level. So if you remember, within an individual atom, you have the nucleus of the atom, you have electrons present in shells, right, around that, okay, so we have some atom here. Some electrons will get promoted, is the idea is. That an electron, say here, will get pushed up into an outer shell. It's an excited electron It's very unstable, doesn't really want to be there, it's not supposed to be there, and so it'll drop back down to its ground state where it's supposed to be. But that energy now that is there, that's been absorbed, okay, so it absorbs the light energy. The energy is used to excite the electron, but as the electron drops back down to its ground state, we call it the ground state, energy can't be destroyed. It has to go somewhere, right? So the energy is released. And in general, what happens is in this structure we call the antenna array, we can think of it in a way like a funnel, okay? A little tornado type thing. And there's all these pigments that are part of the antenna array, ones that are absorbing red wavelengths of light and blue wavelengths of light and yellow wavelengths of light and so on. And they're all channeling this energy down to a particular pigment molecule. A particular pigment molecule that is a chlorophyll A molecule. And that chlorophyll A molecule is called the reaction center. Okay, reaction center, that's, we're going to label that number 2. So 1 is antenna array, 2 is a reaction center. The reaction center is always a chlorophyll A molecule, one very specific pigment molecule of chlorophyll A. There can be other chlorophyll A molecules in the pigment that are not the reaction center. So all chlorophyll A's are not reaction centers. All chlorophyll A molecules are absorbing light at certain wavelengths as well. Some of them can be part of the accessory. But there's a particular chlorophyll A molecule who has all the energy funneled to it. This one who's the reaction center. And something slightly different happens there. The energy, instead of being shuttled on, if this is the, not just any old pigment, okay, but if this happens to be the reaction center, chlorophyll A, when light or the energy from all these other electrons, okay, so it's not electrons themselves, it's energy from them. So when I first was learning this, I remember I had a very difficult time because I was confused from many pictures and books and other places showing what seems like the electrons are being passed from these other pigments to the chlorophyll A. That does not happen. It's energy that's passed to the chlorophyll A. Okay, this is just energy from the dropping of the excited electrons to excite the electron of a chlorophyll A. So not electrons, but just energy from electrons. And then what happens is this excited electron is taken. It's removed from the reaction center. So the reaction center becomes oxidized. It loses an electron. That goes to something called a primary electron acceptor. I'll give it a different color here just so we can see it. That's going to be number 3. A primary, that's a number 1, little prime, electron acceptor. And this is going to be the 3 parts of a photosystem: the antenna array, the reaction center, and a primary electron acceptor. So the antenna array are all the pigments absorbing the light energy. They funnel, they get excited, but then the energy drops down. They don't lose any electrons. They do not become oxidized. Okay? They just pass on the energy, they get their electrons back. But the reaction center, when its electrons get excited, they don't drop back down. They stay excited and they're removed in the excited high energy state. And so now this guy here, this primary electron acceptor, now has gained an electron, and it's a high energy electron. That's going to be the beginning of now light energy has been transformed into chemical energy in the form of a high energy excited electron. So light is essentially exciting electrons, and we get an electron that's excited and then start to move it to do something. Now, how it moves is going to be somewhat similar to, if you hopefully have already studied the mitochondria and you studied the electron transport chain. You remember in the electron transport chain, in the mitochondria, there was a membrane, and embedded in that membrane were proteins. Those proteins passed along electrons from one to another to another. The electrons dropped from high energy to lower energy to lower energy to lower energy, eventually going to oxygen. That's kind of what happens in the electron transport chain. And as the electrons are passed along, there's a transformation. Energy goes somewhere. It's used to do active transport. It pumped protons into a space. They became high energy. That was the storage. Then they diffuse out of that space, and that made ATP. Well, the thing is, that's going to happen here as well. Right? So this electron, one of the things that it's going to be used for is making ATP. Not directly. You'll see that again in a lot of pictures. So this makes ATP. No, what it does is exactly what the electron transport chain in the mitochondria does. It powers an active transport pump that's going to pump protons. They're going to accumulate into space. They'll diffuse out of that space. And then an FOF1 ATPase, just like in the mitochondria, will make ATP. I'm going to go into this in detail, but that's kind of the overview, okay? And again, if you've studied this in mitochondria, then you followed what I said. If you didn't study it yet, then you're kind of like, I don't know what you're talking about. That's okay, because I'm going to explain it, what actually happens there in detail. I'll go over it slowly so you can actually see, and we'll draw some of those pictures. So right now I'm just going to finish this up with where we are as kind of the beginning introduction to it. And then what we're going to do is focus on the light reactions, or what we call the light-dependent reactions. We're going to go over the two different photosystems, photosystem 1 and photosystem 2, the names of the different intermediate molecules and how where they are in this case. So there's going to be two photosystems. Each photosystem is going to have an antenna array, a reaction center chlorophyll, and a primary electron acceptor. How are the two photosystems? So we have photosystem 1 and photosystem 2. Photosystem 2, often referred to as P680. Photosystem 1, referred to as P700. The overview of this, I'm going to draw this out again separately and stop this lecture and then move on to a new one to begin with this. But right now, as we end this one, what's going to happen overall is this. In this membrane here, which is kind of this membrane kind of continuing on here, we're going to have electrons from, and this is kind of weird, but it's from photosystem 2, from the P680 one. So those electrons, the excited electrons, are going to be passed to the primary electron acceptor. The primary electron acceptor is going to give those electrons to an electron transport chain, which will go into details. There's the names of plastoquinone, plastocyanin, a B6F complex that is the active transport pump, and so on. We'll get into the details, but right now, just the idea. The electrons are passed on to here. This will give the electrons to photosystem 1. Photosystem 1 will take those electrons to replace the ones lost by its reaction center, because its reaction center was oxidized. It'll then give those electrons ultimately to a carrier called ferredoxin. That'll give them to another enzyme, but ultimately they're going to be carried by NADPH. So again, hopefully you've studied metabolism already in cells and glycolysis, pyruvate oxidation, citric acid cycle, and so on. You know the main product of those that we follow is the NADH, which is an electron carrier. Here in the chloroplast, we have NADPH, which is pretty much just like NADH, very similar, just the same kind of thing. One thing you'll see as a generic overview or sort of summary is that these light-dependent reactions will make two things. We'll see that they make NADPH, and we'll see that they make ATP. You'll be like, you say, well, where's that? It's like electrons going through an electron transport chain to another pigment, and who then leaves the pigment that goes to NADPH, and it stays there. Well, this electron transport chain, remember I said it's going to pump protons into a space. They're eventually going to diffuse out of that space through a separate protein, who's then going to make ATP. All right, so that'll be separate. So, but it's an indirect part of this, but it's kind of part of this as well. One thing you should take note of is this. Every time this reaction center loses its electrons and gives them to the NADPH, it can regain new electrons from this electron transport chain. It's going to be feeding it electrons. And it loses an electron, gains an electron, gains an electron, and it's just going to keep moving like that. Back here, our P680, our origin, it loses electrons, and the question might be, well, what happens to it when it loses the electrons? Is it destroyed? And it could be, but we have some things in place to prevent that from happening, or plants do, or and photosynthetic cells do, to say. And what they do is they use water. So they split water, all right, into hydrogen and oxygen. Now, what happens there in that process is that the hydrogens are then split into protons and electrons. The protons are then put out into solution as hydrogen ions, and the electrons replace those lost by P680. So when we originally had an equation for photosynthesis, and you had water and carbon dioxide equal, the sugar molecules, the very generic one. The carbon dioxide is going to come in later. That's going to be what's going to make the organic molecules. Then we have the water. We said, where does the water come in? Well, the water comes in essentially here, and I'll go back over this again in more detail as I blow this up into the full sort of view, the detailed view. But the first photosystem, which in this case is called photosystem 2, it might be easier if you just remember it as P680 and P700 because the numbers are backwards. 2 comes before 1 in this particular case, just because of a naming issue and the timing of their discovery, really. So P700 was discovered first, and then when they found a second photosystem, they just referred to as photosystem 1 and 2. And they didn't know that they were connected or that there was a particular order to them, but they were acting independently. And then eventually we discover that, oh, no, they actually connect to each other in this process, and it just happens that the second one discovered is where everything begins. But for that particular photosystem, its electrons are constantly supplied by water. So essentially, if you don't have water, then this process would stop, right? Because it's not going to get electrons to power anything else. So again, this is the second step in talking about this. We're looking at the structure of a chloroplast: outer membrane, inner membrane, thylakoid membrane, 3 membranes. Spaces, we have the intermembrane space, the stroma, and then the thylakoid space inside them. At the membrane of the thylakoid is where we have all the reaction centers, we have all the pigments. This is where the photosystems are located. The photosystems are all the pigments, the antenna array, the specific pigment called the reaction center chlorophyll, and then a primary electron acceptor. That will take the electron always from the reaction center chlorophyll and pass it on. Within the 2 different photosystems, how are they different? One will pass those electrons on to NADPH. The other will pass those electrons on to an electron transport chain. So our next step is to blow this up into all the details and look at the individual molecules along this path. This is all considered light dependent, okay? Because without light, this doesn't happen. This would shut down. We're going to then look at the second part of this later, which is called light independent, which is going to rely on 2 things. Those 2 things are going to be ATP and NADPH. That means the other reactions can happen during times when it's light, because there should be ATP and NADPH. And they also can happen when it's dark. When the lights go out, they'll still continue as long as there is ATP and NADPH. So what we're making here is what's going to power then the reactions that will then pull the carbon dioxide out of the atmosphere and actually start to build the bonds to make the precursors to the sugar molecules. Right? So that's kind of the summary here. Make sure you kind of have the overall layout of the structures and the terms.